within this chapter, and then we'll talk about a couple kind of big points. The big points that we're going to address are really the actual enzyme modeling, okay, so more complex enzymes. In chapter 6, it was substrate, inhibitor, enzyme. There weren't any allosteric effects. We don't have secondary interpretations. Chapter 7, we go on, well, now a lot of mechanisms have this secondary effect. When a substrate reacts with it, it can activate it so that it reacts more. Okay? Or an inhibitor can bind and it reacts less. So it starts to change and it doesn't fit those same dynamics that we looked at with our competitive, non-competitive, uncompetitive situation. Okay? So we'll talk about 2 new models. Both of those are going to revolve around this idea of active versus inactive enzyme. And then for some reason, you're going to look at 50 billion one-letter codes for each of these stupid things. Okay? It's silly. Then we'll look, there's 2 sections. In my opinion, it's kind of piecemealed. We'll look at some kind of interesting effects, so phosphorylation and this Zymogen effect. And then the very end of it is ultimately the organic chemistry behind enzyme reactions. Okay, so we'll look kind of back towards chapter 3, 4. Chapter 4, we had protein structure, our primary, secondary, tertiary structure. And then we'll talk all the way back into organic chemistry, what bonds are actually exchanging, what are the roles of individual amino acids or substrates, okay? Very superficially. Okay? So, what's the deal with allosterism? Okay? I meant to check this, but I think I forgot to check it to verify. But allo, you're looking at a change, and sterism, you're referring to your shape. So allosterics is ultimately how does our shape change? And when we're looking at an enzyme, how does the enzyme shape change? All right, through the course of an individual reaction. And this was something, I think, Stephanie, I think you asked on Tuesday on what was going on with allosterics, and I said I needed to check because every place I've seen it, at least initially in your textbook, has been in reference to a positive effect. You could also have a negative effect. Okay? So allosterics is really just referring to a change in the shape of your overall structure. So that can happen with both increasing the reactivity or decreasing the reactivity. So we get these 2 possibilities coming out of this. That positive effect can be done by an activator, or it can be done by the substrate itself. Okay? So if you have a couple different binding sites for where your reaction occurs, that means multiple substrates can bind into it. So if we bind one, we can now increase the binding affinity for our substrate in the other. Have we seen that before? Hemoglobin. Okay? Hemoglobin is looking at this positive allosteric effect. Actually, I'm not sure if it's allosteric, but it's definitely a positive effect. Okay? Then we could also look at a negative effect, and in those cases, we're just looking at an inhibitor. The inhibitor, I guess it could technically be the substrate too, I can think of some cases. Something binds to our enzyme, changes the overall structure, and it now prevents the binding of something else. Okay? So we've got a nice overall graph of what we've looked at before. We've got our velocity, our reaction velocity versus our substrate concentration. In blue, we've got no secondary effects, no activators or inactivators, or activators or inhibitors in our reaction. If we take a look at the red line, what ended up happening is we put in something that activates the enzyme to react faster. So what happens? Okay, we increase the slope on our activity, and our rate speeds up much faster. What happens when we put in an inactivator? Okay, now it drops down, and we can notice a couple effects. Not only does the line drop down, but what else happens? What does it start to look like? It looks like Vmax dropped off too. Okay? So we've got a couple different things we can evaluate, because that's not necessarily the case. At least the way this is drawn, that's what we see. So we can look at a couple different of enzyme effects. These can get classified into 2 systems, our V system and our K system, okay? Or a mix of both of them. What are those referring to? V system is looking at something where the enzyme max velocity is changed. K system, where your Km is changed. Okay? How do we figure out our Vmax and our Km? We can look at the intercept. So we could look at a Lineweaver block. Lineweaver Burke plot with that straight line and calculate it off of that. Can we get that same information off of this curve? Where's Vmax on this curve? At the very top. So we can determine our Vmax. It's where it plateaus. Okay, so that does give us our Vmax. Can we get Km? Right? So Vmax we can interpret. Km we haven't talked about how we can pull this one off. Actually we can get the Km off of these curves. Km is also known as K 1/2. Why is it K 1/2?
Half the substrate required for
No, it's not the substrate. You were right initially with the first sum. What happens if we look at half our Vmax? What if the rate is half of our maximal rate? What happens at that value?
substrate equals K.
We go through and look at our curve. We can go through and substitute, okay, and I'll just initiate this. If we say our V is actually 1/2 Vmax, we can now go through and solve this or start to rearrange our terms. And what we'll end up finding is exactly as Chuck just suggested here. Km will equal the substrate concentration. So given this curve, can we determine what the Km is? Well, do we know Vmax? Vmax is where it plateaus. Can you calculate half of that value? Yeah. Okay. Now what we need to do is match that up to our curve. Do we know the substrate concentration at that point?
Yeah.
What does that tell us? Km. So we can interpret Km and Vmax off of a nonlinear curve. Okay, so what happened to Km with the activator?
Shifted.
Which direction?
Go to the left.
Fair enough. Bigger or smaller?
Small.
It got to a smaller Km. What happened with the inhibitor? Well, that one becomes actually a little bit more complicated. If we assume that is our Vmax, half Vmax, we're looking in here. I'm going to have to actually mark that. So half, so that would be our Km for our inhibitor. If we looked at the Km for our other one, roughly half. Oh, take a look at that. Came out as roughly the same. Okay. And pretty close considering I really just eyeballed almost all of that. Okay. So the Km may not necessarily change. In this case, the Km didn't change with our inhibitor. It only changed with the activator. Okay. So we can interpret relatively quickly some of the big changes that can happen off this. We don't have to go to that straight line plot. We can interpret it off of these curves. Okay. Questions about that? So I want to make sure that you're aware of that extra little addition for finding our Km, or at least approximating. So changes with our shapes. Okay. As you might very well guess, this is going to drastically change our kinetics, or our kinetic models, and the mathematics ultimately behind them. And I know as much as we all loved that last section with all the kinetics and the algebra and all the numbers changing and shifting and with all that fun stuff, thankfully we don't have to look at that again. So what you're going to be responsible for looking at that standard curve that we just showed an example of, where we're looking at our rate versus our substrate. I don't know why I do a new, or our R. Okay, and interpreting that curve and what's kind of going around that. We don't have to deal with that linear system anymore or how to get it into the equation in a straight line. Okay. There are two primary mechanisms that were developed to explain these observations of allosterism. Okay. Those two methods are your concerted method, or your concerted model, and your sequential model. So what they're ultimately representing is how we can get these shifts around between the different structural changes. Okay? Both of them are centered about the enzyme, and this is where we start to bring in these one-letter codes. We have the active form of the enzyme, and we have the inactive form of the enzyme. The active form is referred to as the relaxed form and given the acronym R. Okay? The inactive form is referred to as tight or taut and is given the term T. So what we're looking at is an equilibrium between these two systems. Okay? We've got the active form and our inactive form, and how the enzyme exchanges between these two states is going to have a drastic effect on the overall kinetics, okay, or the speed of that reaction. All right? So we're going to be referring to R and T. I'm going to try my best to keep them straight. Okay? Can't guarantee that, but that's what I'm going to try. So R is our active, T is our inactive. Okay? And yes, we will bring in some new letters here in just a second. So first we'll talk about our concerted. We've got that active versus inactive state. In the active state, okay, our R form, it's going to bind our substrate rather tightly, okay, which is what we would need to generate products, okay, or have a relatively higher substrate affinity. Okay. When that substrate binds, it's going to change the structure to favor more binding of substrate. Okay. So in the drawing that we've got set up here, what they're showing is an overall enzyme. Let's just use blue, why not? Okay. We've got our overall enzyme in that R state, and what we're saying is that our substrate, which we'll see the shape for in a second, okay, is, I think I even colored that appropriately, a little blue box, fits rather nicely in that slot, and we now have a conformational change once it binds to further activate the other binding site for more substrate. Right, and yes, we'll look at a slightly better diagram in a second. Okay. The inactive form is first off known as the native state. Most enzymes stay in this inactive state. So when we look at the equilibrium, which side do we favor if the T form is our native state? Favor the right-hand side, our T form. Okay, so if we were to evaluate, go back to kind of the nomenclature we used in 152, an equilibrium constant, we'd be looking at our T form over our R form. What size for our K value would we expect? Large. Okay, because most of our enzyme is in that T state. Okay. Would have been nice if they'd used standard notation, but instead of referring to that as K, what do they refer to it as? And we bring in another letter. Now we're looking at L. Okay. So L is effectively the equilibrium constant between those two states, okay, or at least related to that. Okay. In the T form, the substrate does not bind very well. Okay. So if we looked at our enzyme, most of the time it doesn't bind our substrate in the case of the allosteric enzymes. When an inhibitor does bind, it's going to change the structure of our T state to further disfavor substrate binding. Okay? So with this mechanism, we're really concerned about an equilibrium between these two states. So what happens when the substrate does end up binding, okay, to the R form? Okay, well, now it's no longer R, it's R plus substrate. So what happened to the concentration of R if substrate binds? It goes down. We're no longer at equilibrium. What happens? We shift our equilibrium to form that missing R. Okay, so as substrate starts to react with the enzyme, we can replenish with fresh enzyme, and we end up speeding the reaction along. So we have now where our R and substrate is bound, which is now further activated, and then we have refreshed R that can now bind another substrate and become even more activated. Okay? So you're looking at kind of that balance around that equilibrium. Okay? What would we, let's see, I'm going to cheat, see what I just changed this slide around. And my internet changed. Oh no, we can leave it like that, that works. So L shows our balance of this inactive to active relationship. What happens as L gets bigger? Valid point. Let me ask that question differently. If we looked at a graph of our substrate concentration versus our rate, and we had just our standard enzyme on there, what happens if L gets bigger? We're going to expect less product to form. Okay, it's going to become slower. So what we end up seeing with our L getting bigger is that you get lower and lower lines. I hope I didn't swap that one around. Okay. We also have this other term that they decided to bring in here, this C value. Remember I said it's an alphabet soup of letters. Okay. C is looking at the ratio of the binding effectiveness between our R form and the substrate and our T form and the substrate. Okay? So it's Kr for our binding effectiveness for the R form over Kt. Okay? The binding effectiveness of our T form. What happens is that changes. Okay? And I think that one becomes a little bit trickier. Spaghetti works. Good, I did the L right. Good. Okay, you could go through and say spaghetti, but what we're concerned about here has to do with the effects of the allosteric binding. Okay, so let's take a look. So here's our version with our L. As L gets bigger, we end up seeing less steep max velocity, or less steep initial rate. Okay. What happens when C starts changing? So our blue line is a C of 0, and our purple line is a C of a 0.1. Okay. We could potentially look at a Vmax, but we're going to be more concerned about what's happening at the other end of this. What's the big difference at the other end? Okay. How fast it initially starts out. In the case of our purple line, it starts out initially very, very fast. And so what we could actually approximate in our purple system is that we have first order and a zero order. So no really allosteric effects. Okay? It doesn't change its rate as we bind substrate. What happens when we look at the blue line? We're getting that sigmoidal shape is how we're referring to it, that S shape. It's becoming more and more allosteric. Okay? How do we get a C value close to 0?
Really small number divided by a really big number.
Really small versus a really big number. Okay? If we have a very small Kr, what we're saying is the substrate doesn't bind to our R form very tightly. Okay, well, if it doesn't bind very tightly, okay, we aren't going to get a whole lot of change in that one way or the other. Okay? Kt, so that means as we bind more substrate, there's no binding affinity. So as we put on more, there's still no binding affinity. What happens with Kt? Okay? Kt needs to become really, really, really big to get C closer to 0. Okay? If Kt becomes really, really, really big, what we're saying is that the substrate binds pretty tightly to our, these are dissociation. These were their dissociations, right?
Because if KT
Yeah.
goes to KR.
Kt gets really big. What ends up happening, where are we? We get closer to our 0 value, and we get that sigmoidal shape. Okay? Meaning that as we bind substrate, we start to change the overall substrate affinity. Okay? And we start to shift more into that R state. Shit. It may be right, but that doesn't mean it looks like you understand what I'm saying, so I don't know that that counts. And unfortunately, I don't have a better way of explaining that at this point. Okay? So what we're concerned about is that as C gets bigger, okay, we start to become less and less allosteric. The smaller C is, okay, the more allosteric our binding is. Okay, so let's just stick with that. We can worry about explanations on why that is at a later date. So what we're concerned about is how it's changing our overall plots. Kind of makes sense? Okay. So the better graph of this is when we zoom out a little bit further, and instead of looking at just our R and our T states, okay, which is what we just looked at, if we bind our substrate, what ends up happening is we shift out of that R, sort of out of that R state. We've reduced the concentration of our R in our equilibrium, which means we shift to form more of our standard R. And at the same time, we increase the binding affinity for more substrate with that new state. Okay, so we get almost a dual effect in this case. We can produce more R, which binds substrate better, and we also have a new R structure that also binds substrate even better.
Could you say it's almost analogous to a positive feedback?
Yes. Sort of. The positive feedback is a bit odd in that respect. But it's a positive effect.
You're feeding it more
Okay.
and more.
You're feeding it more and more. We see a positive contribution to this. Okay. As we put in more substrate, it favors that substrate formation even more, or product formation even more. If we put in an inhibitor is going to do ultimately the reverse on this. It's going to take our native T state out of commission, which means we're going to have to shift our equilibrium to form more of that T state. Okay, so we end up reducing the amount of active enzyme to react to form our product. Okay, so what we're concerned about in this case is when we have 4, 5, 6 equilibria, okay, all in kind of a balance with each other. And depending at those individual states or individual concentrations, we see slightly different effects in our plots. Okay? So rather advanced system on what's happening here. What you're responsible for is realizing we're at a balance between these two states. Okay? If we activate it, we've now effectively removed the activated enzyme, which shifts our equilibrium to form more enzyme or more active state that can then propagate to our product more. Okay? If we add an inhibitor, the inhibitor removes our T state, our equilibrium shifts to form more of the T state, and we end up reducing the rate of our reaction. Okay? So it's kind of what's going on with our concerted mechanism. I'm sure you all got tons of questions on that, right? All right, at least hopefully. What's that? It does seem a little bit odd as far as this goes. And it also doesn't fully explain a lot of the enzyme reactions that we see, which is why we have other mechanisms. Yeah.
I'm having trouble grasping what makes it determine what state it's in. Because I understand the...
So our enzyme period is always going to start in this R or T state. Okay? And there might be better to specify them as slightly different. Because if I say R, I'm referring to this R, not these Rs.
Okay.
So we've got R0 and T0. So our enzyme is always in a balance between those two states. It favors naturally that T0 state. If we add an inhibitor, we get rid of T0.
We push more to R.
Okay? An inhibitor binds to T0 and converts it into this, for lack of a better term, T state. Okay? If we look back at our overall initial equilibrium, what happened? T0 concentration went down. Which way does our equilibrium shift? It's going to shift to make more of that T0 state. Okay? More of that T0 could now potentially react with an inhibitor and generate this T state. Okay, so we get a T0 and a T state, for lack of a better term. If we look at the reverse on it, okay, without the inhibitor, we put in an activator or our substrate. Okay? The activator can come in and bind with the R0 to make our R state. What happens to the concentration of R0 if the activator binds?
We don't have any R0.
R0 decreases. What does our equilibrium do to account for that?
It wants more
it's going to convert some of the T0 over to R0. We now have more active enzyme that can now react to form products. Does that kind of make sense?
Yeah, that makes sense.
So you do see this kind of allosteric effect because as you put in activator, you generate more active enzyme. As you put in inhibitor, you generate more inactive enzyme.
So as the left side decreases, the right side
Will shift to account for that equilibrium.
does that also pull from the T state? Like, well, affect.
From the T versus the T0?
So like a
I
chain reaction.
don't know on that. These are technically all equilibrium. For it to pull from the T state, we'd have to kick out that inhibitor to get it to shift up. And I don't know in that case. I think what we're concerned about is primarily that equilibrium. Okay? And it's going to achieve an equilibrium with whatever concentrations are there. Eventually, the inhibitor will come off. Why would the inhibitor come off?
Just
How is the inhibitor bound?
not
Not covalently. Okay, we're looking at intermolecular forces, which means that inhibitor can come on and off. As soon as the inhibitor comes off, we're back to this T0. We need to account for that equilibrium. So in our concerted state, we're really concerned about just the primary equilibrium between R0 and T0.
and no actual effects.
And we don't necessarily see an effect on the outside. Okay. So with that inhibitor, because remember, the inhibitor can come on and off indirectly. Just because I increase the concentration of this doesn't necessarily mean it's going to spit the inhibitor off. Okay? Because they are all in equilibrium, you are going to see a contribution there. How much that contribution is important would get into the mathematics behind each of those equilibrium steps. What I'd prefer you to focus on is the equilibrium between our R state and our T state. Okay, our R0 and our T0. Does that make sort of sense? So don't start bringing in the side equilibriums. Focus on that primary equilibrium between R0 and T0. Other questions on it? Hi. We also have a slightly different version. Okay? In this case, we get this sequential allosteric enzymes. Okay? To me, this image looks a lot more simple, but I think that's going to belie the complexity and the mathematics behind it. Okay? What we're looking at is ultimately our enzyme existing primarily in a T state. Okay, just as we discussed with the concerted mechanism. So our T state is our primary native state. And so what's going to happen in these cases is that as we bind an activator or our substrate, we start converting that T state into an R state. Okay, so let's take a look at our very first version of this. So let's ignore the bottom. So I'll cross that out temporarily. So we start at our T state, and substrate binds. Okay, you'll notice that it doesn't have a very high binding affinity, at least based on the picture. Why might you say that?
The shapes don't match.
Here we go. Let's go back to hopefully as preschool as we can get with this. It will make my life a little bit easier too. Okay. Our shapes don't match. So we don't have a high affinity for our substrate. Eventually the substrate will bind. Okay, it can happen. Remember, everything is thermodynamic. Everything's thermally moving with the energy already provided based on our temperature. Okay? So all of those shapes are potentially variable. Once our substrate comes in there, it's now going to change that thermodynamic effect. And what ends up happening is we will slowly start converting our T state into an R state. Okay? But it's only going to be one part of our enzyme necessarily, and that's what they're showing in this case. So we end up converting half of our overall T molecule into an active site, because now we can produce product with the substrate bound, and we'll still have this inactive portion. Okay? With the substrate binding, we have this allosteric effect that then changes the T portion. The T portion now can convert over into our R. So we take our overall enzyme with ultimately two T states. Substrate binds, okay, initially relatively weakly. That substrate then causes one of our T states to change its shape to convert it into now an active enzyme. That active enzyme now has a steric effect on the other half of the enzyme, converting it into an R state that it can now produce product as soon as substrate binds. Okay? We would have increased the affinity for our substrate, okay, once we've done this complete conversion, because look at the site. Okay? The site in our final state matches the shape. So now we don't have to worry about this thermodynamic fighting ultimately between those two. Well, I guess it's on the other side of it. The thermodynamic arrangement of those individual molecules to force the substrate in there. It's now already set its active site to say, hey, I'm ready for the substrate as soon as you bring it by. Right? What we're seeing in this case is what's known as a positive effect, or positive that word. Okay? Which I think I spelled right. It's a positive effect because as we bind substrate, we increase the affinity for more substrate. Okay? This one at least looks simple on the surface because we don't have to deal with all these weird equilibrium shifts all the way through, because it's kind of a stepwise or a sequential model. Okay? But what happens if we put in an inhibitor? In the case of an inhibitor, we still start with our inactive T state. The inhibitor comes in and it's going to bind, and in the process, we now change that from our T0 state to our now inactive T state. That induces a structural change within the enzyme that causes a shift in that other case to prevent binding. Why did they just duplicate it? Oh. Our inhibitor binds, changes the conformation of our T state to another inactive T state. Notice that the shape does slightly change. Okay. That change in shape causes the other half of our enzyme to change its shape, preventing the affinity for our substrate. So as we put in inhibitor, we see these kind of sequential changes in the structure, which then stack to end up preventing more substrate from binding. Yes?
I don't know if it's just a diagram kind of thing, but how come that looks like the T that the substrate comes to has the binding site for inhibitor, but the T that the inhibitor comes that looks like has the binding?
Good question.
Isn't that just like a diagram thing?
I have no idea. I looked at that and wondered that too. I think their diagram is pretty crappy.
A biologist drew it.
So basically, in a nutshell,
No response.
The substrate just makes it, you know, more
The substrate makes it more active.
active, and the inhibitor makes it more active.
So basically
More
shutting
inactive.
down the whole entire enzyme. when
Yep.
you inhibit it.
Yep. So with our allosteric enzyme, we have multiple substrate binding sections. So we could use hemoglobin. We've got those 4 potential sites. Right. As we bind more substrate, the structure changes to activate those other sites. If we have an inhibitor, the inhibitor comes in and changes the structure to inactivate those other sites.
Makes sense.
Okay, so you're looking at a balance of all of those sites initially having the same potential lack of reactivity. Once we force a substrate in there, we can now increase the activity of the other sites. Once we have an inhibitor in there, we decrease the activity of all of our sites.
Makes sense.
Yeah.
So initially, when you put in the inhibitor, are you also saying that it's binding tighter to the inhibitor? That's now the allosteric changed it, correct?
It does change the conformation to allow for more inhibitor to bind.
Okay, so you can technically
So-
make that enzyme even more.
Less
or
active.
less active. essentially,
Less active or
by
more
changing the
inactive. Yep.
Okay.
Okay. The bonus of this, if you want to call it a bonus, of this model is that what we get in this secondary case is what's known as negative cooperativity. Hey, I actually said it. Okay. Whereas in our previous mechanism, with our concerted mechanism, we don't have that negative effect. We can't predict a negative cooperativity. We don't see that negative co-op effect. Okay.
Like, it's gangster.
Questions on that? Okay, so now we got to go through and look at the mathematical build behind how do these enzymes. I'm kidding, Victor. We won't look at the mathematics behind that, thankfully. All we're really going to be responsible for there is recognizing what those pieces are and how we get those general differences between those. Okay, so our primary difference between our concerted and our sequential is that we get the negative co-op effect with our sequential. Okay. After that, we start going through a bunch of extra other things that we could go through to change the substrate structure, be it either through addition of substrates or inhibitors or actually covalently changing the structure. I'm afraid to ask. What's your question?
I was gonna ask if this is kind of like meta, ortho, and para binding.
Interesting question.
Like I'm just trying to find something to relate it to.
Kind of. If we went through and, so this might be a stretch, see if you guys remember this. Yep.
These are ortho
What's the problem with this reaction?
Any of those would still be an inhibitor.
What was that again?
What's the problem with this reaction? Very common side effect of this. What's the product? See if you guys can nail that part
You
down.
get a
Oh, it is like that. So, once it binds, it keeps, so like it will go all the way.
That's our first product.
Oh.
What's the issue with this product?
Because it's
You already have that methane over
This
there.
is an activator.
Yeah, so it won't allow you to bind.
So typically when we do, anybody remember these reagents?
Yeah, man.
This reagent was known as the
No.
Oh, I don't-
Friedel-Crafts alkylation.
Freedom.
The issue with our alkylation reactions is we further activate our product, or we activate the benzene ring for more substitutions. Okay, so you could actually relate it back to the ES. It does work in that case. We could go through and do deactivating reagents as well and do the same kind of idea. So we could look at our methyl group as almost this positive cooperative. I should have just not stopped. I think I had it. The positive co-op effect. As we bind our substrate, what ends up happening? Our reaction, or our enzyme, if you will, becomes even more active to put more pieces on.
Yeah.
Okay? So it does relate back to the ES.
I was just
I like your question.
That's cool.
This has been an improvement from Tuesday. And not on your part, on my part, from liking your question more. Okay, so we can relate this back, particularly for those of us that are organic-minded in this respect, because we do have that kind of cooperative effect. Okay? So can we go through and do something else to the structure to activate it? One of the more common activating groups is this phosphorylation. It can work as an activator, it can also work as a deactivator, but the examples that they picked in the textbook are primarily for activation. So what we're looking at, well, let's go through and ask these questions first. Why might phosphorylation be involved in enzyme activity? Where's
You're
the
asking more of the bonds of when you
Just-
basically phosphorylate, you having more energy put into the system?
where are we getting the phosphate from most typically?
ATP.
What is ATP?
Energy.
A source of energy. Where's that source of energy? The phosphate group. Okay? So if we're going to look at enzyme activity, putting a phosphate group on is usually a pretty good sign of that because that's where a lot of our energy is stored. Okay? So by changing the structure, by putting on or taking off that phosphate, we can now add energy into our reaction to allow it to occur quicker or slower depending on what we're looking at. Okay? So it's kind of an almost obvious thing on why phosphorylation would be involved in enzyme activity, because it is a big source of energy. Okay? What amino acids do you think would be prone to this activity? You can kind of cheat and look at the picture up above.
Eating very polar.
Serine residues. Why would serine residues be active for phosphorylation? What does our phosphate look like ultimately? I wrote LG, but in this case we're really looking at ADP. which is the equivalent of Lg. What does Lg mean? Leaving group. To do a phosphorylation, what happens? We're doing effectively a substitution. Our phosphate, the phosphorus, acts as what? I think I heard someone mumble it. An electrophile. So if we're going to do a phosphorylation reaction within enzyme activity, what amino acids are going to be good for phosphorylation? Any amino acid that can act as a nucleophile. Okay? So serine works. What other ones could potentially work?
The one with the phenol.
The one with the phenol on it. Tyrosine. Tyrosine could work. We got to be a little bit careful with the acidic ones. Okay, the acidic ones do have an oxygen with the negative charge, but is that oxygen as nucleophilic as, say, something else? Not as much, because that negative charge is resonance stabilized within the carbonyl. Okay. Yes, it can happen, and it changes the overall, technically, the functional group. It just needs to get pushed a little bit further along. Okay. Are they only activators? No. Okay. Remember, ATP is a common source of energy. We can use that to either activate or deactivate something. Okay. Why might that be important? Are all of our processes going one direction? No, we have the reverse. Think catabolic versus anabolic. Okay. So we do have these opposite direction enzymes. So we could actually put something on there to activate the catabolic pathway, but at the same time, if we put that on an anabolic enzyme, it's going to push it the other direction.
So this helps that.
Zymogens is a separate issue. We'll talk about that in the next one. The next big kind of key word out of this is kinase. So it's in reference to enzymes that allow for this. And what we're looking at is the catalyzation of the phosphate transfer. Okay. Why would we group that as a big group? Why is it not just one kinase that does it all?
Because there's more than one kind of kinase.
Why is there more than one different kinase? What different structures? The phosphate's the same in every single case. The amino acid is different. Okay? If that amino acid structure changes, the enzyme that has been designed specifically to wrap around that amino acid isn't going to be able to wrap around another one. Okay? So we will see a variety of different kinases depending on what amino acid or where you're trying to phosphorylate. Okay? Questions on your phosphorylation? Okay. Zymogens. So all a zymogen is an enzyme that is physically inactive. Okay? It's not the T state. Okay? The T state was our inactive enzyme, but it had the potential to be active. A zymogen is physically inactive. It cannot function even if the substrate comes along. Okay? And that's because there is something wrong within the covalent structure preventing the proper folding of that enzyme to allow for its activity. In the case of our active versus inactive, we're looking at forces. There's a difference in intermolecular forces within that protein inactivating it. Okay? The zymogen, it's a covalent linkage that's preventing that. The example that we'll keep coming back to is chymotrypsin. So chymotrypsin in its natural state, okay, or chymotrypsinogen, that one out too, is our zymogen state. You'll notice that when we take a look at this, they're trying to show that it's all linked together. We've got, assuming this is the entire thing, 245 amino acid residues, okay, all pumped out in the primary structure. What ends up happening is that this long chain of amino acids gets spit out into the digestive tract, and enzymes like proteases will go through and start to cleave particular amino acid connections. That first particular amino acid connection is between 15 and 16. That cleavage frees up now the ability for this structure to refold. We've broken, instead of having a nice long chain that has to now fold up and maintain that linkage, it can now freely rotate and come around somewhere else. That structure is now partially active. Okay? So what we've done is changed the covalent structure and converted it from a zymogen, which is inactive, to now an active enzyme. This active enzyme can now catalyze a self-digestion at another position, Okay, they're listing it off here. Oh, a couple different ones actually, it looks like. Where we end up cleaving out these two other pieces. Okay, so we break the connection between amino acid 13 and 14, and we break the amino acid connection between 146, 147, and 148 and 149. So those pieces are now no longer covalently bound, which means they fall out of the structure. What we've again done is opened up a section where now we have these three pieces that can fold around each other, and we now have our uber active alpha chymotrypsin.
So it's like cascading.
Yeah. Okay. So in each of these primary structure changes, we can induce a secondary structure change.
Are
Okay.
you looking at 3 different enzymes at this point?
These,
or are you looking
these
at
are all
a
the same
quaternary structure?
enzyme. Okay, so all we're looking at in this case is technically the primary structure. That blue bar is supposed to represent all the amino acids in that sequence. In the very first step, okay, did I list it up there? Trypsin, there it is. Trypsin comes through and cleaves the primary structure. So now instead of it being one peptide chain, or polypeptide chain, it's now 2. Okay. That cleavage now activated this portion of the enzyme that allows it to then go through and cleave these other bonds. And now we've got a new form of the enzyme that actually has 3 peptide chains not directly linked to each other, sort of. Okay, we'll talk about, they're not linked to each other through a peptide backbone. That's true. We've broken the backbone there. Does that kind of make a little more sense?
So the enzyme's kind of holding all these pieces.
So then how do all these pieces stay linked to each other? That's ultimately looking at our tertiary structure. So what things contribute to our tertiary structure? Go ahead and say whatever.
But they don't necessarily need to stay linked together. They could be new enzymes in themselves or whatever they're
The active enzyme has these 3 peptide chains
Those aren't enzymes.
linked. So this overall thing is the enzyme. Okay. If I remove one of those, it's not the enzyme anymore.
Because you've got the 3 different pieces, you're looking at a
So the
quaternary
enzyme is
structure, because you've got 3 proteins.
Yes.
In this picture, the enzyme is the green line?
No.
No, it's not.
So that brings up a good question. What's the, and I think you're saying yellow.
Green from back here.
It could very well be. The screen does look a little pale. Okay. Those are yellow-green lines. Okay. What are those yellow-green lines doing? You'll notice that in each of these cases, they don't change.
They're holding it together.
That's how they're
How?
twisted around and
You're right. These lines are holding it together. What is specifically doing that holding?
Like the nitrogen, the disulfide.
Disulfide bonds. What we've got are disulfide bonds holding these things together. So we have to show these weird linkages like this. Okay, you'll notice they're all different lengths. Why are they different lengths?
Because they're binding different spots, like twisted differently.
It's all back to the tertiary structure. Remember, to form the disulfide bond, they have to be next to each other. We have to have a sulfur in one case and a sulfur in the other binding. What 2 amino acids? Cysteines. Okay, because we have that thiol on it. So why do we have these longer lines? Because what we're showing here is the primary structure. Remember, our proteins can fold up on each other, and just because amino acid is bound, 1 is bound to 2, doesn't mean amino acid distance-wise 1 is really far away from 243, because it can fold back on itself, and that ends up partly that's what's happening in these cases. Okay, so those linkages are what's keeping the structure all together. We're ejecting those other pieces from it, and we now have our active enzyme that can go through. So ultimately what we're concerned about is that we can generate some polypeptide chains that are very close to what our final enzyme will be, but they've got too many pieces. We have to eject those pieces somehow. Okay? In that non-ejected state, we're looking at the zymogen. As soon as we go through and cleave those unnecessary pieces, we can now activate our protein, and we have our active protein that can function. Why might we want to do that?
so it doesn't tear up whatever before it gets into the digestive tract.
Yeah, we could look at a release of this perhaps chymotrypsin is generated, but we don't want it to be active right away until it encounters a particular thing that it should destroy. At that point and that point only do we want it to become active so it can interact with it. Okay? So we could have this polypeptide chain floating around just waiting to get activated or put into its active form so that it can actually catalyze that reaction. But maybe that reaction would also destroy a bunch of other things.
Destroy the cell that made it.
Chymotrypsin is also a protease, okay, also known as a caspase. Their roles are to cleave proteins. Okay? So it may be a really good thing to have in your digestive tract so you can get the amino acids out that you want. Is that something you particularly want floating around in your cells or your bloodstream? No. Probably not a good idea, because that's going to start destroying everything it encounters, not just the food that we're trying to get out. Okay? So we want to be able to transport these things to the appropriate spots before they start destroying everything. Yeah?
Do we, is it always an enzyme activating? Is that like so an enzyme activating another enzyme?
To do this, yes, because we're looking at physical bond cleavage. Actually, I should change that, because we could run acid-base reactions that could cause a bond to cleave as well. So it doesn't have to be another enzyme that does that cleavage.
Are they all like, do they all like have like cooperativity, like, you know?
Not necessarily.
Like pepsin, you know, like pepsinogen.
So
It has cooperativity.
I keep meaning to look it up and actually get the image of it, okay, that summarizes all of biochemistry. And it's this massive tangle that looks like somebody just vomited up a bunch of spider webs, okay? And that's ultimately biochemistry. Every one of these enzymes is tied to probably 20 other enzymes, okay? And what we're trying to focus on is keeping it as simple as possible and slowly start building together these other pieces and how they can interact with each other. At this point, really all we're concerned about is going the basics, overview, and then when you really pushed into a particular biochemical pathway, you would focus on just that pathway and say, how do these enzymes interact with each other? What things are needed to make them react? Okay? Like sources of ATP. Okay? ATP was necessary in that previous case, okay, as a phosphorylation agent. Where did that ATP come from? It didn't just exist, it had to be produced by another enzyme. Okay, so a lot of these products will tie across to each other. Active site chemistry. So what we're ultimately looking at here is what's going on in the active site. Okay, so we've got a bunch of things that we could potentially consider. We've got the primary structure. So what amino acids are directly involved where the chemistry is occurring in our active site. We could also look at the secondary and larger structure, because the secondary and larger structure is going to be what holds our very particular amino acids in that active site. Not only that, but it's also going to, be what holds our substrate in the active site. Okay? So we need to be aware of both our primary and larger structural considerations. We can then also look at the actual mechanisms that the enzyme is undergoing. And we kind of talked about that with our phosphorylation. The alcohol in serine is acting as a nucleophile with the phosphorus electrophile within ATP. Right? How did those two get near enough to each other to allow for that reaction to occur? It was likely an enzyme, okay, that helped force those two close to each other to allow that reaction to occur. Okay? So that's kind of our organic mechanism and beyond. After that, we could look at specificity, which ultimately brings up the subject of chirality. I'm not going to talk about it a whole lot, but this image I think works pretty well. Remember, most enzymes, biological enzymes, are designed to catalyze one very particular reaction. Okay? Trypsin is used to do the cleavage of, I already forgot what a bond was. Trypsin is used to cleave the bond at arginine. Okay? So it's very characteristically looking for an arginine next to a certain sequence of amino acids, and it will cut that bond and that bond only. For it to go through and do that, it has to have a very particular structure that allows for that active site, okay, to bind that specifically. So a lot of biological enzymes tend to be very, very, very specific because they want to be used for one very particular purpose. It's not always the case, but it does show up. Why has chirality become important? What kind of amino acids do we have?
Oh,
We only have the, okay, since you're all saying L, I'll accept that as the answer. I actually didn't know. I don't remember which one it is either, but we only have one particular set of amino acids. Okay? If we go through and look at DNA, which well, I guess we haven't officially talked about, our DNA tends to form primarily one type of helix. Okay? Yes, there's slight variations off of that, but we tend with one type of helix. We have a chirality within that helix. So virtually everything in biology has a chirality or has a handedness associated with it. If our enzymes are going to catalyze those reactions, they must also have that handedness, okay, which means they can only work in a very particular fashion. Do we have any lefty-handers in here? How do you like right-handed scissors? Okay, anybody use left-handed scissors? Okay, how did you like using the left-handed scissors? So we use scissors as effectively our enzyme to allow us to go through and cut things. Okay? Scissors were primarily created for right-handed people because most people are right-handed. Okay? If we use left-handed scissors, it tends to hurt our right hands. Enzymes are very similar to that function. They have a particular handedness. They cannot do the other method. Okay? And we'll end up using chymotrypsin again as an example. So for chymotrypsin, what it's catalyzing is the cleavage of an ester or peptide bond. We're primarily concerned about the peptide bond. Why the peptide bond? That's how proteins are built, is through peptide bonds. So it tends to be rather selective for the peptide bond. But the ester bond is very, very similar in chemistry, so it can also cleave ester bonds as well. Okay? And it also tends to be selective around these two amino acids, our phenylalanine and our tyrosine. So what does that necessarily mean? Some part of that enzyme is looking for that aromatic ring before it can do the cut. Okay, so you can think of that almost as your thumb and forefinger. So, primary structure. What amino acids are going to be critical to that reaction? Kind of a hard question to ask, at least superficially. So what we would end up doing as biochemists is we would take the enzyme and we would say, okay, I'm going to start changing amino acids. When does the enzyme stop working? Okay, so we selectively change one amino acid at a time to see if it changes the overall reactivity. If it doesn't change the reactivity, then that enzyme or that amino acid was likely not involved in the active site. Okay, so when we do the changes, we also want to make sure that we're doing the changes in such a way that we don't drastically change the structure. Because if we change the overall three-dimensional structure, it's going to lose the reactivity, whether or not our primary acid or primary amino acid change was in the active site. Okay, so we might swap alanine for I could see that. We could go alanine for valine. We're looking at hydrophobic interactions and relatively small shapes. So we're trying to do amino acid matches as best as we can all the way through it. Kind of makes sense? I was going to go with glycine, but valine is probably pretty close. Okay. So this is why, in theory, you're supposed to have all those amino acids memorized. The other option that we can go through and do is we can label particular amino acids. So we could probably go through and do a first approximation by doing these amino acid swaps on it. And then we could also go through and change the structure of amino acids. So we can put tags on them. The two tags that were used in the case of chymotrypsin were the DIPF, which reacts with serine residues. So that now neutralizes serine's reactivity. We could also go through and put on the TPC or toothpick for a histidine residue. Also, it's going to limit histidine's reactivity. So those substitutions on the amino or the primary amino acid structure, I was right, we can use to determine what amino acids are critical in the active site. So it turns out that histidine 57, so the 57th amino acid in the polypeptide chain, and serine 195, so the 195th amino acid, are both in the active site. What does that mean? Protein had to fold back on itself to get those two close to each other. Okay? So we now have an idea on the two critical amino acids in the reaction. They have to be involved in the mechanism. Okay? Really wanted to finish this, but I don't think I'm going to. So I guess I'll go ahead and stop it there. Secondary structure, we'll pick up with that, is how did those amino acids get near each other? How does it fold back on itself to get those back?